So let us discuss about the floral induction development and the genetic and molecular analysis. This part is your related with your genetics that is given on your flower gene development section. I hope you will able to understand what I am going to be taught you this time. I will going to teach you right now. So let's get started now. So about the floral induction and development, let us see what we have to study in this part. In this part, we will have a brief introduction and then the flowering time genes that will influence the duration of the vegetative growth of the plant, and also we will study about the flower identity genes and the overlapping of these genes in duration of time for particular function. So how do they work? Let us begin. So let us before that we have to study the structure of a flower, a bisexual flower. Well, you all know at the beginning part there is sepals. Okay, then comes the petals. Okay, then comes the stamen, which is your male reproductive part of the flower. The stamen consists of anther and filament. Within the anther, there are pollen grains. There are pollen grains that is the what can I say, sex cells of the plant, male flowering plant. Well, we are studying about bisexual flower. The female part of the plant together called as carpel. The female reproductive part of the plant is called carpel, which consists of stigma, stele, and the ovary. So, this is the general structure of a bisexual flower. Now, let us study in detail. Well, it has been said here that flower development provides a model system to study the mechanism that govern the pattern formation in plants. So, based on the genes, what are the genes that are responsible for the flower development? We can study them as a model system to see that what are the factors that influence the gene to form that flower and how the flower is formed, because so much of structures are present within the flower: the sepals, the petals, the ovary. What come first, the sepals come first or the ovary come first? Okay, based on that we scientists become interested in studying those genes that causes the flower development. Even the fruit development genes are also being studied. Okay, which genes come under first influence and what interaction occur with the other genes to make the flower development? So, some scientists have proposed various models for studying the development process in flowers. Now, as you can see in the figure that most of the flowers consist of four organ types that are present in specific order. Now, it is present in specific order. Okay, first comes the sepals, the ground part, the base part. Then comes the petals. Then comes the stamens, and then comes the carpels. To in if we from above, if we see from cross section, if we see first the sepals come, then the petals come, then the stamens come, and then the carpels, which are the female reproductive part of the flower, and stamens are the male reproductive part of the flower. Now, obviously, it's not like that petals come last or sepals come first or stamens come in second way. Not like that. It is quietly regulated. It is quietly in a regulated manner they are being developed. And we all know that all these things are developed by specific genes. These are the organs of the flower which are specified by some genes that are interacting with one another, which we have to study in these notes. This is the main thing that we have to study in our syllabus here, that how the sepals are being formed, how the petals are being formed, what genes interact to form stamens, to form carpels. That is the main thing that we have to study here. Okay, so here we are not considering, we are not looking at all the flowers. We have to take a model organism for the flower development, and the most common organism for studying the flower development is Arabidopsis thaliana. Okay, generally for animal study we use Drosophila, the fruit fly. Here in case of for flower development we use Arabidopsis thaliana. Arabidopsis thaliana are small flowering plant whose genome size is also very short, and the life cycle of Arabidopsis thaliana is quite short, so it is very good to study the genes responsible for their floral development, for the flower development. As their life cycle is short, so quickly they will bloom, and thus we can able to study more and more about the genes responsible for the development of the flowers in Arabidopsis. Okay, now generally it is saying that the flower development is initiated when a signal arrives at the shoot apical meristem. So within the shoot tip or shoot apical meristem, which consists of meristematic cells, there from there a signal molecule triggers that shoot apical meristem for the development of flowers. Actually, that signal is actually allows that apical shoot meristem to trigger the genes that are responsible for the floral development. Okay, so mainly the flower develops from the shoot tip, or you can say shoot apical meristem. In short, we say SAM. Now, when a particular signal, now there are different type of signals that can takes place to initiate the flower development within the shoot apical meristem. Now, shoot apical meristem is made up of meristematic cells. During the vegetative state, the shoot apical meristem is programmed to produce leaf primordia. What is vegetative state? I think you have forgotten, so I am trying to uh, recapitulate that term. Vegetative state refers to the phase of plant growth as written here. Vegetative state refers to the phase of plant growth that occurs after germination or seedling stage and before flowering. So, just before flowering, just after germination and before flowering, between that stages is called the vegetative state. So, vegetative state refers to that stage of plant growth where flower development has not occurred but have arise just after germination. Now, during this time, during vegetative state, plant bears most of the leaves. So, plant develops most of its foliage or plant leaves. At this time, during vegetative state, strong root system also occurs. The tissues of growth, that is the meristem, are busy in producing leaves. Okay, so this is the part where we know that vegetative stage is that stage where the plants are actually growing their uh, leaves, growing strong root system, strong vascular system. Okay, and they are busy in producing the leaves. Now, what it is saying that leaf primordia arises from the shoot apical meristem, that is from the shoot tip. The leaf primordia arise during the vegetative growth of the plant. So it is saying that during the vegetative growth of the plant, leaf primordia arises from the shoot tip, from the shoot apical meristem. And this leaf primordia consist of group of cells that will form into new leaves. Okay, whereas the flower primordia are the little buds that we see at the end of the stems, from which flower will develop. The flower primordia arise due to certain signal which triggers the shoot apical meristem to activate some genes which will lead to flower development actually. Okay, so I think you have understood what I have tried to say. Now, when the floral signal arrives from the leaf, suppose a signal molecule arrives from the flower primordia from the leaf, then the meristem acquires that floral identity, and the secondary inflorescence meristems or floral primordia arise in the axis of the uppermost leaf primordia. So this is the quite a flow chart that I have tried to draw. I have tried to write. You can write in big. I have write in short. So during vegetative state, the shoot apical meristem triggers the growth of leaf primordia. That is, from the leaf primordia, new leaves will develop. Then what happens? A signaling molecule appears, and that signal molecule trigger the shoot apical meristem for flower development. Okay, then that shoot apical meristem gets triggered by the signal molecule and induces the floral primordia development. The floral primordia developed on the uppermost part of the leaf. From there, your flower will develop. Okay, so let us see more about that flower development. So, just I said here, so during the vegetative state, it is that phase of plant growth that occurs just after germination, just after your seedling stage and before flowering. During the vegetative state, the plant develops most of its plant leaves, strong root system. Okay, and now if it is a vascular plant, then vascular bundles will also develop due to the influence of some signaling molecule. Okay, like for instance, some hormonal signal. For example, that signal molecule may be a flow region that you will study just now, that you will start going to be studied now. I will be studying you just now. This signal molecule actually induces the meristem. It actually triggers the meristem to induce the formation of floral primordia. Okay, and then from that floral primordia, which will be developing on the uppermost part of the plant. Okay, from there, so the floral primordia, wait for a minute. So floral primordia actually they are the little flower buds that we see at the end of the stems from which the flower will develop. And and they are the floral primordia will trigger the genes that control the flower development. So in this way, your whole process is occurring. Now we will study mainly about the flower developmental genes, the genes that regulates the flower growth. Certain genes called flowering time genes influences the duration of the vegetative growth. Flowering time genes provide connection between florigen or floral induction signal and transition to the floral organs. So floral induction signal, the signal molecule is called as florigen, which causes to be transferred from the your floral primordia to the other floral organs to induce the growth of the flower. Well, floral induction signal or florigen, which is a flowering hormone, which is like a molecule that is responsible for controlling or triggering the flowering in plants. So this is that main signal molecule which induces the flowering development genes to do their function. Okay, and florigen is also found in the leaves. It acts in the shoot apical meristem. Normally this florigen is normally found in the leaves. It is produced in the leaves, and it acts on the shoot apical meristem, that is, it acts on the shoot tip of birds and the growing tips, that is the growing tips where the genes will act. It will provide that connection. So this signal molecule is mediated by the genes, the flowering time genes actually. So the flowering time genes provide that connection, provide that linkage between florigen to the floral organs, which will develop into floral organs actually. So again I repeat, florigen is actually a floral induction signal molecule which is used for controlling or triggering the flowering in plants. Florigen is produced in the leaves and it acts on the shoot tips, that is in the shoot apical meristem of birds and in the growing tips through the help of the floral flowering time genes. This florigen is going to be transferred, that provides that connection. Now suppose if there is mutation, change in the base pair. Suppose if there is mutation in the flowering time genes, it may interfere with the production of signal in the leaf, translocation of the signal to the shoot tip, or its activity in that shoot tip. Apex means shoot tip. So if the flowering time genes which provide that connection, the florigen or the flowering hormone, which is actually responsible for controlling or triggering the flowering in plants, suppose if that the genes which responsible for conveying this florigen to the shoot apical meristem, which is called the flowering time genes, if that flowering time genes get mutated, then it will interfere with the production of signal in the leaf. That signal will not be able to trigger in that site. There will be no translocation, means movement of that signal molecule to the shoot tip, because by the way I have already said that florigen attacks on the shoot tip, on the shoot apical meristem, and it also act on the growing tips with the help of the flowering time genes. So due to the mutation of the flowering time genes, the flowering time genes will not mediate the function, and thus there will be delay in the flower development. Okay, and most of the flowering time mutant genes are identified, thus it causes the plant to flower later than the normal, indicating that the mutant can interfere with the pathways that normally promotes flowering. So the gene responsible for flowering, we will see them one by one in detail. So these flowering time genes, if they get mutated, it causes delay in the flowering, as I have said, and that indicates that mutant form of flowering time genes can interfere with the pathways that leads to the flower development. So the flowering time genes and the flower development genes, they are playing an important role in growth and development of the flower at particular time. If a mutation occur in that gene, the flowering time genes, which is providing that signal, that florigen or flowering hormone signal to the shoot tip, it will be delayed, and thus there will delay in the flowering development. For example, I have said about Arabidopsis. We are using Arabidopsis here as a model organism for flower development. For example, Arabidopsis species, it is a facultative long day plant with a critical photoperiod of 8 to 10 hours. Now, two terms here to be mentioned here that I have provided in the notes. What is photoperiod and what do you mean by long day plant of 8 to 10 hours? Photoperiod or photoperiodism is a term which means that the amount of light and darkness a plant is exposed to. The amount of uninterrupted darkness is what determines the formation of flowers on most types of plant. It is saying, again I repeat, don't be confused. Photoperiodism is a term which means that what is the amount of light and darkness a plant is exposed to for the formation of flowers. The amount of uninterrupted darkness will determine the flower formation of flower on most type of the plant. So depend on the darkness. What is the amount of darkness that will determine the flower development in most of the plants? That is what photoperiodism means. Okay, but you will feel quite awkward. How is it possible? Plants make their food during photosynthesis, so it's due to the daytime that affects the plant to make their food. So why not flowering? Because at the daytime pollination occurs, right? But that's not always true. Some plants bear flowers at night. They bloom at night for the pollination done by the nocturnal animals, the night animals, bats, owls. Okay, some insects are also there. So here photoperiodism simply means what is the amount of light, what is the amount of darkness a plant is exposed to, and this amount of darkness will determine the formation of flowers in most type of the plants. Previously, botanists used to think that the length of the daylight a plant was exposed to will determine that whether a plant would form flower or not. But experiments proved otherwise. It is the length of the darkness that a plant experience that plays the most crucial role in flowering. So that's the surprising, that's the awesome, you can say, thing that you have studied now. Okay, that the length of the darkness will determine the flowering of most of the plants. Previously, botanists used to think that it's the amount of daytime that depends on the flowering of the plants, right? But it's not true. It's the length of the darkness that will determine the formation of flower in most of the plants. Now, what is the long day plant? A plant that requires a long period of darkness is termed as it's termed as short day plant. Obviously, if a plant requires a lot of darkness, a long period of darkness, it is called as short day plant. And if a plant requires short time of darkness, it is called long day plant. That means we are saying conversely, reversely. Okay, that means if a plant need long term of daytime light, it is called long day plant. Conversely, we say if a plant require short time of darkness, it's called long day plant. Whereas if a plant require long period of darkness, it is called short day plant. Short day plant form flowers only when the day length is less than about 12 hours. So the short day plant, which are the plants which require darkness for them to bloom their flowers, they require darkness more than 12 hours. That means they require daylight less than 12 hours. Like the spring plants, chrysanthemum, chrysanthemum, chrysanthemum. In Bengali, we call chrysanthemum as Chandramallika. In Hindi, we say them as Guldaudi. Guldaudi. I think I have correctly pronounced this term, Guldaudi. So chrysanthemum is such type of short day plant. Then poinsettias, poinsettias, which are the plants, flowering plants grows in America, in Mexico. Then Christmas cactus. These are the some examples of short day plants. If these plants are exposed to more than 12 hours of light per day, then flower formation or blooming will not occur. So they require more darkness for them to bloom, right? Called as short day plants. Whereas other plants require only short night to bloom, which are called as long day plants. Okay, so other plants require only a short night to bloom, which are called as long day plants. And these are termed as long day plants because they require more than 12 hours of sunlight. Like most of the summer blooming flowers, garden vegetables are long day plant, right? So it's quite interesting that we have studied about this long day plant and short day plant. And what's the main objective of our study here, that I have said that most of the flowering time mutant genes are identified, which that causes the plant to flower later than normal, which indicates that mutation in the flowering time genes can interfere with the pathways for the flower development. Okay, now we have to say the genes, but I'm trying to make that concept clear. Okay, it is interesting to note that although large number of late flowering mutants have been described, no single Arabidopsis mutants that remain vegetative identity has been found. So mutant genes has not been observed till now. This fits with the general assumption that there are multiple pathways that control the flowering time with a certain amount of built-in redundancy. So again I repeat, it is saying that although there are there may be mutation in the flowering time genes which can interfere with the flower development, but no single mutated flowering time genes in Arabidopsis thaliana plant has been found. So that's conclude us with this point that there must be multiple pathways for flower development. There is not a pretty straightforward pathway like glycolysis like that. There are multiple pathways for flowering development, and thus, even if mutation occurs, it will not leads to interfere with the flower development. Okay. Okay, it has also been said here that there are multiple pathways that control the flowering time with a certain amount of built-in redundancy. Now, what is redundancy? It is also called as genetic redundancy. Genetic redundancy means that two or more genes are performing the same function. So there must be let us complete this part, okay, then I will discuss this paragraph, this sentence. Genetic redundancy means two or more genes are performing the same function, okay, and that activation of one of these gene will have little or no effect on the biological phenotype. It means to say that there are multiple pathways for flower development. So even if one of the flowering time genes get mutated, there will be no problem because there are multiple genes which are carrying out the same function. That is called genetic redundancy. So genetic redundancy means two or more genes performing the same function. Here the same function is flower development. That inactivation of one of these genes will have little or no effect in the biological phenotype. Here, the biological phenotype we are discussing about flower development, right? So, this concludes that there are multiple pathways which are controlled by multiple genes and they are interacting with one another for the flower development. So, even if a single mutation occurs in one of the flowering time genes, no problem, other flowering time genes are there which are being used in other pathways for flower development. Now, in Arabidopsis, we have seen at least there are 5 separate but interacting pathways for controlling the flowering time that has been identified. Now, I will go to the details about the flower development genes. In Arabidopsis thaliana, scientists have revealed that there are about 5 separate pathways, and these 5 separate pathways are also interacting with one another. That means one pathway is interacting with the other pathway for controlling the flowering time, which has been identified by the scientists. Let us reveal that pathway. Flower or floral development genes are those genes which are helpful in the development of different floral organ, and this is mainly occurs due to expression of some genes. So the flowering time or the flower development genes, there are many type of genes for flower development which are helpful for development of different floral organ, the stamens, the carpel, the petals, the sepals, right? And these genes are organ specific, so their expression leads to the development of different organs of flower like sepals, carpels, stamens, petals, etc. So they are interacting with one another for the development of different floral organs like sepals, carpels, stamen, petals, etc. So when these organ specific genes get expressed, only a complete organ of a flower will develop. Now let us see that flower development genes. So generally, if I say the flower development genes, well, I have discussed in the next page. Okay, there are mainly four. There are mainly four classes of flower development genes. So floral development genes are of four classes, which are again further divided into many classes. But for simplicity, I will try to discuss them in a simple way. I will try to discuss them in a simple way. Okay, let us see those flower development genes in a typical flower. Here we have used the model flower of Arabidopsis thaliana. Let us see. So these are the different flower development genes. The first one is your what? Floral meristem identity genes. The first one is your floral meristem identity genes. Now these floral meristem identity genes are class 1 floral development genes. The function of these floral meristem identity genes is the transition of a shoot vegetative meristem to floral meristem. In simple words, we can say that these floral meristem identity genes are helping to convert the shoot part into floral part, into flower part. Okay. So floral meristem identity genes are those genes which are needed for converting the shoot part into flower part. Initially, we used to see that the shoot or the vegetative part of the plant are expressing. But when these specific genes means when these floral meristem identity genes are going to be express themselves at specific locations, at specific locus, then only conversion of shoot to flower occurs. So again I repeat, floral meristem identity genes are what? Floral meristem identity genes are those genes which are needed to convert the shoot part into flower part, or you can say transition of the shoot vegetative meristem to floral meristem. Okay. Initially, we know that the shoot or vegetative parts of the plants are going to be expressing just after germination. But when these floral meristem identity genes are going to be expressed at specific location, then only conversion of shoot to flower occurs. So there are total 3 floral meristem identity genes. There are total 3 floral meristem identity genes. And what are those 3 floral meristem identity genes? I will write them one by one. So these three name of 3 floral meristem identity genes are: number 1, leafy. We say them as leafy, L-E-A-F-Y, in capital letter, in short we write L-F-Y, leafy. Second is Apetella 1, Apetella, Apetella 1. In short we write AP1. And the third floral meristem identity genes is your cauliflower, C-A-U-L-I, cauliflower, F-L-O-W-E-R, cauliflower gene. We, in short, we write them as cal, C-A-L. So these are the examples of class 1 floral meristem identity genes. Well, this leafy gene that has been mentioned here, this leafy gene activates the Apitella 1 gene. The leafy genes actually activates the Apitella 1 gene. If the leafy gene gets mutated, then instead of flower, we are going to see the structure of leaf. Okay. So This much you have to remember, that leafy gene activates the apitella 1 gene here, and if the leafy gene gets mutated, then instead of flower development we are going to see the structure of a leaf. The cauliflower gene are expressed in early stages of flower development. So these are the examples, these are the genes which fall under the category of class 1 floral meristem identity genes. The second is your shoot meristem identity genes. Let us see the shoot meristem identity genes. Well, where it is written, we have to see. Well, the shoot meristem identity genes is the opposite. This gene has the opposite effect that of class 1, if I am not wrong. Wait for a minute. So we get class 3, we get class 4. Okay, no problem. Okay, so these are so this example is the class shoot meristem identity genes. So the shoot meristem identity genes, the shoot meristem identity genes has the opposite effect that of class 1, and it maintains the identity of shoot meristems when expressed. So it is not helping in flower development, but it is regulating the development timing of the flowers. So this gene, the shoot meristem identity genes, has the opposite effect that of class 1 floral meristem identity genes. The shoot meristem identity genes maintain the identity of the shoot rather than flower. So if the shoot meristem identity genes get expressed much higher rate, then your flower will not develop. Only shoot will develop because they are maintaining the identity of the shoot meristem. And it is expressed, it represses the flower development. It is not going to express the flower development, rather than it will going to repress. It acts on the shoot apical meristem. This shoot meristem identity genes going to be act on the shoot tip. That is, it is going to be act on the shoot apical meristem to repress the floral transition and maintains the vegetative growth. It prevents the transition of inflorescence shoot into floral shoot. Okay, so it prevents, it maintains their vegetative growth, the normal growth without any flowers. That is, these shoot meristem identity genes are preventing the transition of inflorescence shoot into floral shoot. So it is not going to develop into floral organ. These genes are meant are regulating the expression of the floral organs. It is trying to repress the flower development. Example of such the gene responsible for the shoot meristem identity genes is your TFL, one of the genes. Well, TFL is of two types, TFL1 and TFL1 and TFL2. Well, what is the full form of TFL? The full form of TFL is terminal flower. The full form of TFL is the terminal flower. Okay. So just now I have said about the Class 2 shoot meristem identity genes. It is repressing the development of flowers. It maintains the identity of shoot meristems when it gets expressed, and it is the opposite of class 1. Uh, it is the opposite of class 1 floral meristem identity genes, right? It prevents the formation of floral transition, okay, and maintains their vegetative growth of the plant. So we have done two of them. The third class, which are involved for flower development, is your floral organ identity genes. Floral organ identity, floral organ identity genes. What does it do? Is these genes, these floral organ identity genes, mainly controls the floral identity. That means the all organs that will compose a flower, the sepals, the petals, the stamens, then the female reproductive part, which are called as what? I have forgotten. Carpel. Okay, the carpel, right? So the floral organ identity genes mainly control the floral identity. That means it maintains all the organs that will compose the flower. So they are going to get their identity by the expression of these following genes. Understood? So by the expression of these floral organ identity genes, by the expression of these floral organ identity genes, the organs are going to get their identity. The organs which will form the flower will get their identity with the help of the expression of these genes. So this is quite important. So whether the floral organ is sepal, whether the floral organ is petal or carpel, that will be going to decide by these genes. So these genes plays a crucial role that where the sepals will grow, where the petals will grow, where the carpel will grow, where the stamens of the flower will develop. That will going to be decided by these floral organ identity genes. And wherever they will express, there it will going to act in a particular organ. Okay, so these floral organ identity genes, in short words, they are very specific, they are organ specific. So their expression will lead to the development of different organs of the flower, like sepals, petals, carpels, and stamen. Example of floral organ identity genes are, let us see. On above, I have written Apetella 1, Apetella 2. Well, I have said Apetella 1 as floral meristem identity genes. So why not a one gene can mediate more functions? It may have many functions. So Apetella 1, Apetella 2, Apetella 3, Pistillata, and Agamas. Agamas. These are all examples of floral organ identity genes. And if you see, I have separated these genes in three classes: class A, class B, and class C. Well, it will be required when we will study about the ABC model of flower development. This ABC represents class A, class B, class C. That which genes, which of these floral organ identity genes come first, which comes later on, for proper expression of the flowers, for proper expression of the organs of the flowers, better to say. So these genes are fall under the category of class 3 floral organ identity genes, and all of these genes have two classes that came into existence when we study in Arabidopsis thaliana: class A, class B, and class C. Three classes, sorry, not two classes. And this which classes of for floral organ identity genes going to be expressed, which class of floral organ identity genes will express it later. Okay, that will be decided, that we will study when we will study about ABC model of flower development. Okay, the last class is your boundary setting genes, also called cadastral genes. The boundary setting genes, also called as cadastral genes. Well, this class 4 genes, as I have written here, it regulates the expression of this class 3 floral organ identity genes by limiting their expression. How much they are going to express? What is the area? How much area they are going to be expressed? That will be actually regulated by these class 4 boundary setting genes, also called as cadastral genes. Actually, what can I say? That in Bengali, if I say বাউন্ডারি সেটিং জিন অর ক্যাডাস্ট্রাল জিন নির্ণয় করে যে কত দূর পর্যন্ত ফ্লোরাল ওল যেগুলো আছে, 4 রকম ওল আছে, সেপাল, পিটাল, স্ট্যামেন, স্কার্পেল। তো এই যে ফ্লোরাল যে ওলগুলো আছে কত দূর পর্যন্ত ডিস্ট্রিবিউট হয়ে থাকবে তাকে रेगुलेट करे ए बाउंड्री सेटिंग जीन्स। सो डायरेक्टली यू कैन से हम लोग कह सकते हैं यहाँ पर कि बाउंड्री सेटिंग जीन्स रेगुलेट करता है फ्लोरल ऑर्गन आइडेंटिटी जीन्स को, कितना दूर तक वो एक्सप्रेशन करेगा। Like in many flowers we have seen different types of floral parts, same floral parts but at different their locations are different, right? Somebody have 4 petals, somebody have 2 petals, somebody have many petals like sunflower. So that is decided by the boundary setting gene. So yet actually boundary setting genes रेगुलेट कर रहा है फ्लोरल ऑर्गन आइडेंटिटी जीन्स को कि कितना दूर तक वो एक्सप्रेशन करेगा। फॉर एग्जाम्पल, there are 4 whorls, right? There are 4 whorls in a flower. The sepals, carpels, stamens, and sepals, petals, stamens, and carpels. For example, the third whorl and the fourth whorl, between the third whorl and the fourth whorl, there is a gap. If you see clearly, between the third whorl and the fourth whorl, the third whorl bears the stamens, the male reproductive parts of the flower, whereas the fourth whorl bears the carpels, the female reproductive part. So between the third whorl and fourth whorl, there will be some gaps. And these gaps are maintained by the boundary setting genes. So boundary setting genes are observed on the sides, which regulate the what? Which actually sets the boundaries for expression, for regulating the expression and controlling the correct distribution of the floral organ identity genes. So the boundary setting genes, if I say, they are regulating the floral organ identity genes by setting up certain boundaries for their expression and thus control the correct distribution of the floral organs. So it is regulating the boundary, that how much that floral identity genes are going to be expressed. Like I have said, that the third whorl of a flower is stamen, which are the male reproductive part. Okay, and the fourth whorl represents the carpel, the female reproductive part. Between the stamens and between the carpel, there is a gap, which is actually, this gap is actually filled by the boundary setting genes to separate the third whorl from the fourth whorl. Now, one of the type example of this class 4 boundary setting gene is your SUP gene. SUP gene. Now SUP gene is a simple name, has a simple name called Superman gene. This gene acts to maintain the boundary between third ole and fourth ole. It defines the boundary between the male reproductive organ, which is the stamen, and the female reproductive organ, that is carpel in the flower. So SUP gene is such type of boundary setting gene. An example I am giving here. SUP gene is a boundary setting gene which controls the third ole with the fourth ole. Well, the third ole bears the male reproductive part, and the fourth ole represents the female reproductive part. This third ole and fourth ole are separated by this Superman gene. So they are limiting the expression of these floral organ identity genes that bear stamens and carpel. Okay, so in this way they are limiting the expression. So I think you have able to understood what I have tried to say. So they are actually regulating the floral organ identity genes by limiting their expression, by setting up boundaries for their expression. Okay, and then in this way they are controlling the development of the flower. Okay, so these are the four types of classes of your flowering genes. Now let us discuss the pathways. Let us discuss the pathways. Well, there are many pathways are there. uh, Maybe I can able to finish off these pathways. Maybe, let us see. So let us discuss about the pathways. So there are four pathways almostly which are found in the Arabidopsis thaliana plant. We have studied the model organism here. I am repeatedly saying, okay, and these are commonly found in all plants. The vernalization pathway, the gibberellic acid pathway, the autonomous pathway, and the photoperiodic pathway. So these four pathways are actually controlling the flowering development. And these are controlled by the four classes of genes known as floral meristem identity genes, which are class 1, fall under class 1. Then come class 2, which are regulating the flower formation, which is called as shoot meristem identity genes. Then comes the third class, which is called as the floral organ identity genes. And the fourth, which is called as boundary setting genes or cadastral genes, which is actually limiting the setting up boundaries of the floral identity genes expression to separate the four organs of the flower. Now, all of this pathway that I have here mentioned here, vernalization pathway, gibberellic acid pathway, autonomous pathway, photoperiodic pathway, all of these pathway are linked with one another, and they are merging at a particular point, which will activate the floral meristem identity genes. Okay, which is your LFY, the leafy, and the apitella 1, which further induces the boundary setting genes to regulate the expression of the floral organ identity genes. So initially, your these genes are going to be expressed. Initially, these genes are going to be expressed first. The LFY, the leafy, and the apitella 1 genes. And these class 1 genes are then going to express the floral organ identity genes regulated by the boundary setting genes to be expressed. Well, class 2 genes are not helping in flower development, rather than they are repressing the flower development, right? So in one of time they are manipulating the development of flowers. So again I repeat, initially these genes are going to be expressed first, then it will activate these genes. Well, their expression are going to be limited by their expression These genes expression are going to be limited by these genes, and in this way the flower development occurs. Now, what are the signals which triggers this activation of class 1 floral meristem identity genes that are going to be studied now? Okay, let us start with vernalization pathway. Well, let us discuss about this pathway. So I'm going for the flow chart here. Well, I will try to discuss the notes here. Okay, for simplicity, if you can understand about this pathway. Well, during the cold and chilling period, it leads to the activation of some of the genes such as VIN3, VRN1, and VRN2 gene. So, due to the cold period or chilling period that we are facing now also in winter, it activates these three genes named as VIN3, VRN1, and VRN2. What is the full form of this VIN, VRN1, and VRN2? VIN3 full form is vernalization insensitive 3. Vernalization insensitive 3, the name of this gene. About VRN1, the full form of VRN1 is vernalization 1. Vernalization 1. And obviously, VRN2 means vernalization 2. So during cold and chilling conditions, it leads to the activation of VIN3, VRN1, and VRN2 genes. Now these three genes epigenetically inhibits the expression of FLC gene, which is called as flowering locus C gene. So these three genes, vernalization insensitive 3, vernalization 1, and vernalization 2, are inhibiting the formation of the FLC gene. So it is suppressing the FLC gene. And what does FLC gene do actually? FLC gene full form is flowering locus C. The full form of FLC gene is flowering locus C. This gene is repressing the flower formation by inhibiting the SOC1 gene. So, VIN3, VRN1, and VRN2 are inhibiting the FLC gene. Generally, FLC gene inhibits the formation of flower by inhibiting the SOC1 gene. Okay. But when VIN3, VRN1, and VRN2 is inhibiting this gene expression, automatically the gene, the FLC gene which was repressing the flowering by inactivating the SOC1 gene, are going to be inactivated. So SOC1 gene automatically gets activated because FLC1 gene is repressed by these above three genes, and these genes are actually activated due to the cold chilling condition, right? Now SOC1 gene is actually, it is an activator. It is an activator gene. The full form of SOC1 gene, actually SOC1 gene helps in flower development. How? Let us see. It is an activator gene, and the full form of SOC1 is suppressor of overexpression of CO1. Suppressor of overexpression of CO1. very high name, large name. Suppressor of overexpression of CO1. CO1 means contents. CO1 full form is contents. Now we are not looking at why contents name is given, but this much you have to remember that SOC1 gene helps in flower development. Why? Because it is an activator gene. It activates the LFY gene. And you know the name of LFY gene falls under the category of floral meristem identity genes, right? So SOC1 gene, when this gene gets activated and it activates the LFY gene, which is one of the uh, class 1 floral meristem identity genes, the LFY gene then activates the AP1 gene, which is called apitela 1, AP1, and together it activates the class 3 floral organ identity genes. So both LFY, both leafy genes and apitela 1 genes activates the other floral organ identity genes, which are apitela 1, apitela 2, apitela 3, pistillata, and agamus. Finally, this floral organ identity genes promotes the flowering. I hope you have understood what I have tried to say. Let us discuss again. Well, from the beginning, if I say FLC gene, known as flowering locus C, is repressing the activation of SOC1 gene. SOC1 gene is a important gene for flowering because SOC1 gene activates the LFY gene, which is a floral meristem identity genes, which are required for flower development. But as SOC1 gene is inhibited by FLC gene, so no flowering occurs. What happens during chilling conditions or cold conditions? It leads to the activation of these vernalization insensitive 3, vernalization 1, vernalization 2 genes. So these three genes are going to be activated due to cold and chilling conditions. These three genes are inhibiting the function of FLC gene, and thus SOC genes get released from its repression. It gets activated and it activates the LFY genes. Well, these LFY genes then activates the AP1 genes, the apitela 1 genes, and we know that LFY genes and apitela 1 genes are class 1 floral meristem identity genes, which will activates the floral organ identity genes. The floral meristem identity genes finally activates the floral organ identity genes, which is your apitela 1, apitela 2, apitela 3, pistillata, agamas, which finally promotes flowering. Well, it will be regulated. These genes are also regulated by boundary setting genes of class 4, which is also called as cadastral genes that we have studied a little bit ago. So this is the whole pathway, the vernalization pathway. I hope you have understood this pathway. Let us move on to the next pathway, which is much more easier than this previous pathway. Well, the number fourth pathway is a little bit difficult, which is called as what pathway? Photoperiodic pathway. It's a little bit difficult is there. Let us discuss the other two pathways quickly. Well, I am just going for the diagram, just going for the flow chart. Okay, the next pathway is called gibberellic acid pathway. Well, gibberellic acid is also called as phytohormone. Huh? It is a plant hormone which regulates your stem elongation, which regulates your germination, dormancy period of the seed, flower development, leaf and fruit senescence. So gibberellic acid has many functions. Gibberellic acid regulates the flower development here that we are going to discuss now. It also going to regulate the stem elongation, then leaf and fruit senescence. Senescence is the loss of cell power to divide or grow. Okay, so it also regulate the leaf senescence, the fruit senescence. So now here we will see that how gibberellic acid is involved in flower development. Generally, gibberellic acid is a plant hormone. Gibberellic acid directly activates the SOC1 gene. Okay, previously we have seen that SOC1 gene is activated indirectly by the vernalization genes VIN3, VRN1, and VRN2 by inhibiting the expression of the FLC gene. Here, gibberellic acid directly activating the SOC1 gene, which finally activates the leafy genes, and leafy genes activating the AP1 genes. Together, leafy genes and AP1 genes, the Apitella 1 genes and leafy genes together called as what? The initial class 1 flower meristem identity genes. Okay, these floral meristem identity genes finally activates the floral organ identity genes, which are Apitella 1, Apitella 2, Apitella 3, Pistillata 1, not Pistillata 1, Pistillata, so only Pistillata, then Agamus, and finally it promotes the flowering. So very easy, the gibberellic acid pathway for flower development. Okay, I hope you have understood this pathway. It's very easy. Directly you see that gibberellic acid is activating the SOC1 gene, which finally activates the, it is a activator gene, SOC1 gene, which activates the initial genes, class 1 genes of floral meristem identity genes, which finally activates the floral organ identity genes, which promotes the flowering. Next comes the agamus pathway. The agamus pathway, if I say, with the help of a diagram, if I say, it will be very good for you. Let us discuss. Now, agamus pathway is related with reproductive maturity. In the previous pathways, we have seen that due to chilling or cold conditions, flower develops. Okay, in the second pathway, we have seen increase in the level of gibberellic acid leads to flowering. Well, artificially, we can also induce flowering by this method. The third pathway for flower development is autonomous pathway. Well, in this pathway, it can be achieved when the plant becomes capable for reproduction, right? So when a plant achieve its reproductive maturity, it activates some of its genes, like the flowering control locus A gene, also called as FCA, then flowering locus K gene, then FLD, flowering locus D, and lumini dependence gene, which is called as LD. Lumini dependence gene, also called as LD. Again, I repeat. So, when a plant achieves its reproductive maturity, it activates some of its genes like flowering control locus A gene, also called as FCA, flowering locus K gene, flowering locus D gene, and lumini dependence gene. These four types of genes are inhibiting the function of FLC gene, must same like your vernalization pathway. So these four genes which are activated due to sensing of a plant age, as the plant has achieved its reproductive maturity, these four genes are activated, and these four genes are going to inhibit the function of FLC gene. FLC gene, as I have said, flowering locus C. This gene inhibits the SOC1 gene. Now, as these four genes are inhibiting the expression of FLC genes, so SOC1 genes is going to be expressed now. And this SOC1 gene, as I have said, mentioned in the first pathway, the vernalization pathway, SOC1 gene activates the leafy gene, which further activates the apitella 1 gene. Together, these are called as class 1 floral meristem identity genes. These floral meristem identity genes activates the floral organ identity genes, which are apitella 1, apitella 2, apitella 3, pistillata, and agamas genes, which finally promotes the flowering. Okay, so very easy. In the first vernalization pathway, we have seen that some vernalization genes due to cold and chilling conditions is inhibiting the expression of the flowering locus C gene. Here we see that this same flowering locus C gene is going to be inhibited by the FCA, which is called flowering locus C, flowering locus A, flowering control locus A, flowering control locus K, flowering locus D, and the lumini dependence gene, right? And due to the inhibition of this FLC gene, the SOC1 gene is going to be activated, is going to be expressed now. SOC1 gene then finally activates the leafy gene, which leafy gene finally activates the AP1 apitella 1 gene. Together, these two genes are called as class 1 floral meristem identity genes, which finally activates the class 3 floral organ identity genes, which are apitella 1, apitella 2, apitella 3, pistillata, and agamas genes, which finally promotes the flowering. Very easy. These three pathways are very easy. Now, at the end of this lecture, I will discuss about the fourth and final pathway. In the next video, we will discuss about the ABC model of flower development. Let us see the fourth pathway now, which is quite lengthy, the photoperiodic pathway. Now, to understand the photoperiodic pathway, we have to understand something, the biological clock. Have you know the term of biological clock, also known as circadian clock? Well, biological clock is not like that it will show digitally, but it will makes that living being aware of what is going. Like at the night we feel sleepy, that is triggered by our own biological clock, which is also called as circadian clock. Here it is written, circadian clock. In the same way, plants are also subjected to circadian clock rhythms to promote flowering. Like some plants bloom at night, bloom flowers at night. Okay, if you see that at the morning, early in the morning, the birds chirp, whether it is winter or summer, or if it's a rainy season, that's another issue. But if you see that they are maintaining the same time, it's because due to the biological clock which is occurring in our body. Now, if you are not maintaining that circadian rhythm, one some extra thing I'm going to say, discuss with you little bit about human health and disease. If we are not maintaining the circadian rhythms, then we are going to face a serious problem. Serious disorder may come into our body, like humans are facing nowadays. Humans are working at night also, late at night, overtime, night duty. These are the common things that we do. Or at night we are studying, okay, to make us more intelligent. But that's not an intelligent issue. It is affecting our body. What is the time for us to do? That has to be done through according to this circadian rhythm. Now, Here, according to the plant, circadian rhythm are 24-hour cycle that are part of the body's internal clock running in the background to carry out the essential function and process. Again, I repeat about circadian rhythm. Circadian rhythm are the 24-hour cycles that are part of the body's internal clock running in the background that carry out essential function and process in our body. Not only in our body but also in the plant's body, also in the plant parts also. One of the most important and well known circadian rhythm is the sleep wake cycle. Well, that's not applied to us. It is also applied to us, but we are actually breaking that sleep wake cycle. That's why we are facing problems, complications in our health. But the sleep wake cycle, if you see, if you observe to the other living beings, that's occurring due to that circadian rhythm. Now, this circadian clock is actually triggered by the, or you can say synchronized by the solar time. Okay, so by the according to the solar time, circadian clock is synchronized. Now, if I say, for an example, if you go to outer country, okay, there also you will be affected by that circadian rhythm. For example, you all know that here we are living in India. Today is, now it is night. Okay, now suppose it is night, then on the other part of the world there is day. So if somehow if I go to that place with a quick time, with a short time, then we should not be feel sleepy. We should not be feel sleepy or feel sleepy. It's depend on the solar time. It's depend on the solar, the amount of the sun, at what position is there. It depends upon that factor. Similarly applied to plants as well. In most living things, internally synchronized circadian clock makes it possible for the organism to anticipate daily environmental changes corresponding with the day and night cycles and adjusting their biology and behavior accordingly. So circadian clock is synchronized or regulated with the solar time, and most living organisms including the plants, they are synchronized internally with the circadian clocks and makes it possible for the organism to anticipate daily environmental changes. They can able to observe what are the environmental changes that is occurring. But suppose at this winter night, days are short and nights are long, so we have according to that we have to perform our normal functions, jobs, and all those things are different things. But for providing food and to sleep, how much time, at what time we have to eat, that's the thing that we are talking about here. Okay, so we can anticipate with the help of, by synchronizing circadian clock, by synchronizing circadian clock in our with solar time. Okay, the day and night cycles, and we adjust our body functions, our biology, our behavior accordingly to this circadian rhythm. Okay, so we have talked about circadian clock, that what it is. It is actually an internal body clock which is running all the time till our death, and it will be running in the background, and it carries out the essential functions and processes in the living body. And each time we, with the help of this circadian clock, um, we are actually anticipating, okay, the daily environmental changes that occurs, okay, that correspond with the day and night cycle. Okay, that's why most of the animals, not most, all of the animals, excluding us, uh, they don't get so much of ill. Okay, they remain healthy. Even they get ill, they cured quickly. Okay, but we are actually trying to break the rules of the circadian clock by working all the night. okay, instead of resting. Okay, so let us discuss about this part. We have got to another topic here. Actually, in molecular biology, if I say that circadian clock consists of various transcriptional and translational feedback loops that are conserved across the plant species. If I talk about the plant and circadian clock of the plant, this circadian clock or biological clock consists of various transcriptional and translational feedback loops. That means the genes for transcription of certain genes. Here, the flowering genes. We are mentioning about flowering genes only. We are mentioning about flowering genes here, and the translational feedback loop. That means it's also regulating the expression of the proteins from the genes and regulate multiple pathways like photosynthesis, seed germination, hypocotyl elongation, stomatal movement, flowering, senescence, which is loss of cell power of division and growth. So it is means to say that circadian clock consists of various transcriptional loops and translational feedback loops. Okay, which is which actually conserved among the plant species. So plant species behave according to the circadian clock for the flower development here. Not only flower development, circadian clock regulate multiple pathways like the photosynthesis. Remember the dark reaction and light reaction that you have studied in your junior classes. Also, you have studied in your class 12 also. The seed germination, hypocotyl elongation, stomatal movement, even the flowering is actually regulated by the circadian clock. The leaf and fruit senescence, that means loss of a cell flower of power of division and growth. Now, how circadian rhythm can affect your growth of formation of the flower? Let us see. Well, if I say one by one, in flowchart I have said here, I have not described it. This circadian clock is activating the CO gene. Okay, CO gene, by transcription and translation, it makes CO protein. Now this CO protein, full form of CO protein I have mentioned little bit ago. It is called as contans. CONTANS. This CO protein is stable only in light condition. In light condition. That means in day condition it is stable. At night it becomes unstable and gets degraded. So this during the daytime CO protein, now it is going to be expressed in the phloem of leaves and stem. Again I repeat, this circadian clock, the biological clock, will decide when flower development will occur. Like most of the spring flowers, at the springtime it will develop, it will bloom. So that circadian clock is activating the CO gene. CO gene is going to be transcribed, going to be translated to form that CO protein. CO protein is stable only in daytime conditions, and this CO protein is going to be expressed in the phloem of the leaves and stomata. CO protein going to be activated the FT gene. Well, FT gene is present in the companion cells of the phloem of leaves and stem. Again, I repeat, CO protein going to be activate the FT gene. FT gene is found in the companion cells of the phloem and in the companion cells of the phloem in leaves and in stem. Okay, the companion cells that you have studied besides phloem and xylem, these FT genes which are present in the companion cells are going to be transcribed and translated to form FT protein. FT protein then moves from the leaves to the shoot tip, that means in the shoot apical meristem, and enters to the nucleus and form a complex called FDFT complex. So much critical, so much lengthy pathway. So FT gene activated by the CO protein during daytime. This FT gene is expressed in the companion cells of leaves and stem. FT gene produce the FT protein. FT protein moves from the leaves and it enters to the shoot tip, or you can say it enters to the shoot apical meristem. And within the shoot apical meristem, it enters to the nucleus and form a complex with the FD protein called FD-FT complex. FD-FT complex now activates the SOC1 gene. It also activates the apitela-1 gene. Now, as we know, SOC1 gene is an activator. It activates the LFY gene, which in turn it activates the AP1 gene as well. And together they are activating the class 3 floral organ identity genes, which promotes flowering. So again I repeat, so due to the circadian rhythms, it due to these circadian rhythms, it actually makes the plant to anticipate daily with the environmental changes and to bloom at particular time of the season, right? So due to circadian clock, it activates the CO gene, which is also called as contans. I think I have said contans, CONTINS. CO gene gets transcribed and translated to form the protein, CO protein. CO protein is expressed in the phloem of the stem and leaves. Now it is stable only in light conditions. CO protein activates the FT gene, which is going to be expressed in the companion cells of the leaves and stems. FT gene transcribe and translate to form FT protein. FT protein moves from the leaves and enters to the shoot apical meristem, that is, it enters to the shoot tip, and there in the shoot tip it enters to a nucleus where it combined with a complex forms with FD protein to form FD-FT complex. This FD-FT complex then activates the SOC1 gene, which is activated gene for LFY, and this FD-FT complex also activates the AP1 apitela-1 gene. Together this LFY gene, which is getting going which gets activated due to SOC1 gene, together with AP1 gene, it's going to activate the class 3 floral organ identity genes, which promotes flowering. So that's all for today's lecture. Well, it's quite a big lecture. I hope you have understood. Just go through the whole lecture now. Okay, don't pause and see, otherwise you will miss the concept. Okay. In the next video, I will try to discuss about the ABC model of flower development, that is the class A, class B, class C gene, which is found in the floral organ identity genes. Okay, the floral organ identity genes are divided into three classes: class A, class B, class C. When under class A there is apitela 1 and apitela 2. Under class B there is apitela 3 and pistillata, and class 3C gene represents the agamas gene AG. These three genes are actually interacting with one another to for formation of the different parts of the flower, the wholes of the flower. Okay, one by one. That we will see them in the next video. So if you have any doubts regarding this lecture, you can ask me in the WhatsApp group. Okay.